Hello and welcome to the October edition of the Nutritionist Webinar 2016. I'm Marianne Fessenden from Agricultural Modeling and Training Systems, and I serve as your English language host. These webinars provide access to technical seminars by internationally recognized speakers. The series presentations are held in English, Portuguese, and Spanish. Marcos Neves-Pereira from Universidade Federal de Lavras will be hosting the Portuguese language presentation with Marcelo Hens-Ramos from 3R Lab facilitating. Paula Trillo from Argentina is hosting the Spanish language webinar. There will be a post-presentation question and answer period during which listeners can submit questions through me, Marcos, or Paula. A complete recording of archived webinars as well as a question and answer session for each will be available at the AMTS website. We are very pleased to have Dr. Dave Barbano here today as our presenter. We are additionally fortunate that Dr. Heather Dann will join us for the question and answer period. Dave is a professor in the Department of Food Science. He received his BS in Biology, Food Science in 1970 at Cornell University, followed by his master's in 1973 and his PhD in 1976. He has been in the Food Science Department since 1980, where his research focused on the chemistry and processing of dairy foods, milk analysis, and the effect of farm management practices on milk chemistry. He has co-authored or authored over 200 scholarly publications and is the recipient of numerous awards, including the 2014 Prize of Excellence from the International Dairy Federation. Some of his current research has focused on the use of milk fatty acid composition to guide feeding and management decisions. Heather Dann and the Minor Institute has been collaborating on some of this research. Heather received her BS from Cornell University, her MS at Penn State University, and her PhD from the University of Illinois. She has served as a research associate for Minor Institute in Chazy, New York, and is an adjunct professor at the University of Vermont. Today, Dave will be discussing new frontiers in milk analysis and their effect on farm management practices. Jot down your questions as you listen, and we will ask in the end. Now I'm going to turn the presentation over to Dr. Barbano.
Thank you very much. We're going to be talking today about new frontiers in milk analysis for farm management purpose. And the main focus that I'm going to cover today is on milk fatty acids, but there's much more that we're working on and is coming in the future. I first of all would like to thank the host for the invitation to present this seminar. But secondly, I want to acknowledge the co-authors on this presentation, Melissa Wolpert, who's a graduate student at the University of Vermont, and her thesis research provides part of the information in this presentation. Caterina Melilli from Italy works with me on data analysis. Heather Dann at Minor Institute, Rick Grant at Minor Institute, and Larry Chase, Cornell Emeritus Professor in Animal Science. And it's really been a very wonderful team of people to work with as we've kind of developed and learned how to learn and ferret more information out of milk analysis by analysis of spectral fingerprints of the milk. And that's what I'm going to be introducing to you today in terms of how we're using this in relation to farm management. Current status of precision management is the first topic I'm going to talk about in relation to milk testing and where kind of the state of the art is today in terms of on-farm type of testing. And this relatively new part is not on farm at this point, but ultimately we'll see that the goal would be to try to move in that direction. I'm going to talk a little bit about what farmers want in terms of information to manage and make decisions. We'll give an example of connecting analytical measures in milk analysis to meet dairy farmer needs. And that's where this work that I'm going to talk about is really trying to do things in terms of milk analysis, not just fat and protein and solids or MUN, but to go beyond that in terms of analytical measures which will give us insight into things such as feed efficiency, animal health, reproduction, as we go forward. And then I'll talk after that about some of the future directions in our work, particularly in relation to farm management and sustainability. In terms of precision management milk testing, currently, many of you, I'm sure, are familiar with the Affimilk system. It's a little different than what I'm working on. First of all, it's near infrared, and the technology I'm working with is mid-infrared. The mid-infrared is what has typically been used in DHI type of milk testing labs and for payment testing. It tends to be a more accurate and higher cost way of milk testing, but for payment testing, it's very much justified. What we're doing now with the milk testing is exploring the use of mid-infrared to get other information in terms of useful management information. There's rapid testing that is available that you can do at farms in terms of antibiotic screening for deciding whether a individual cow can go back into the bulk tank after having some treatment, or at the beginning of lactation, the clearing of any dry cow treatment. The mid-infrared for milk components and milk somatic cell count, currently in the U.S., some of the very large farms do have the traditional laboratory equipment, testing equipment for infrared for components and for somatic cell testing that they use to try to diagnose and troubleshoot primarily health and some production problems. These instruments that are used at large farms typically are manual. They're not like the big automated instruments that you see in a DHI lab or a payment testing lab. And that's kind of where things are, at least in the U.S., North America, and where I'm looking to be able to move the technology in terms of mid-infrared and to identify whether we can use it to make management decisions in a more real-time type of application and justify the work and the cost to modify this technology to be able to operate in the long run at the farm, build into the milking system. So that's kind of the longer term goal. But today, the near infrared, the AFI system, it will give you fat, it'll give you protein, you've got your milk weight, but the kind of details we're going to talk about are quite far beyond what you can do in the near infrared. So what do dairy farmers need? They need analytical results that will help them manage the efficiency of feed utilization, metabolic health during the transition period, mammary infection, animal welfare, environmental impact in terms of what they're doing on the farm, the example of MUN and the nitrogen waste going to the environment, but there's other things that we can be focused on too. Reproduction to improve economic performance and sustainability of farms. They need analytical information to make decisions. They don't need a lot of data. They need to make decisions. The success then of farm management ultimately depends on the correct decisions on an animal-by-animal basis, and the challenge is to find what I would call the cow of interest, the one that needs something done and action taken, making a decision and taking an action when we get down to the individual cow level. Ultimately, that's where we'd like to be, but today I'm going to talk quite a bit about the application of this type of milk testing to bulk tank milk and the things we can learn about the herd from the bulk tank. We can learn more from individual cows, but starting at the bulk tank gives us an idea of where this can take us. As farms are getting larger, there's more technology, satellite technology, cloud-based internet tools, information. All of these new tools are becoming available, and there's more of them every day. It's a bit easy to be overwhelmed by all of this in terms of the information, the choices, what technologies, as a farmer, as a producer, particularly as farmers get larger, what things do I need, what things do I not need, How do they work together? That's where it kind of gets to be confusing, and really you get to a point where in the end the milk production is about the sum of the performance of all the individual cows in the herd, and the farmer needs information, not a lot of data and graphs, information that's distilled down to be able to make decisions. And it's very easy to be like the cartoon on the right, where you've got so much data and you're just overwhelmed by the computer and the information, the data that's coming from it, and you can't really see the details of what you need distilled down to action points. So what I'm going to do is give an example of connecting analytical measures to dairy farmer needs, and we're going to focus on milk fatty acid composition. And this is something that I've been directly involved with for about the last 5 years, that has gradually evolved as we've learned how to do it, improve the analytical performance of the methods, and started to collect data in the field in terms of fatty acid composition of milk from individual farms. Our overall vision in this is to develop new tools in milk analysis for bulk tank milk and individual cow milks that will provide information to support decision making for management of feeding, health, and reproduction. Right now, we're focused primarily on the management of feeding. We're doing some things as far as our research and analysis of spectra that will relate to health. Ultimately, reproduction would be of interest, but we haven't really gone very far on that at this point. So in terms of objectives in our work, we first wanted to develop a new rapid analysis tool to measure fatty acid composition, but put the data in a format that's useful for farm management. And this is one of the differences in what we've been doing compared to what most of the data has been from research programs, and trying to get the fatty acid data in a format that comes off an instrument, that's an instrument that you can purchase in a form that gives you insights into the management of feeding. We've worked with the instrument in my lab, and now I've got several of them in the field. They're manual, they're not automated. We have one at Cornell. I've got several collaborator labs now that are working with this that I've set up, and we have our statistical models on them that analyze the spectra. This particular instrument is a Delta Instruments model FTA. They're located in the Netherlands in terms of manufacturing. And this is the instrument we use in my lab and several of the labs I'm collaborating with. The calibration of the instrument for fatty acid measures is done once a month. We produce the reference calibration samples at Cornell. The instrument will test about 50 to 70 samples per hour. It gives you all the normal milk components: fat, protein, lactose, solids, the MUN, or NPN, urea, whatever you want to call it, and all the fatty acid parameters that we've developed all come off the same milk sample at the same time. This doesn't use any chemical reagents. It's a direct analysis on the milk sample. The only thing you have to do is warm it to 40 degrees Celsius. There is an automated version of this instrument that's in the inset down in the lower right of the picture, that the same statistical models that we've developed for the manual instrument run on the larger instrument that can test 600 samples per hour. So everything that we're doing as far as the development of models for predicting other things from the milk actually run on a commercial instrument and can be enabled on the instrument with the support of the manufacturer. The first things that we did relate here in the red on the slide to measurement of fatty acids, but measuring them in groups instead of individual fatty acids. And we broke it into 3 groups in relation to the biochemical pathways that function in the cow that relate to the origin of the synthesis of those different groups of fatty acids. And they are the de novo fatty acids, the mixed origin, and the preformed fatty acids. And I'm going to explain a bit more about that and get everybody clear. Some of you probably know this very well, and others this may be new to you. So we'll go through it, get everybody to the same point. I'm going to first talk about bulk tank milk testing. And our experience so far has been that the fatty acid data in the form that we're doing it, that seems to give us an index of the efficiency of forage digestion, and particularly the de novo fatty acids, which I'll define for you as a group of what's included there in our data, which I'm going to share with you, seems to be a very useful metric to understand how well forage digestion is proceeding in the rumen of a dairy cow. We also have another metric that is called double bonds per fatty acid, and it's a metric about the structure of the fat within the sample. And in our field work, we're starting to see that this appears to relate to milk fat depression. When this number goes up, you're going to be going down in fat, and it seems to be reflecting some things that are happening in the rumen that correlate with milk fat depression, and we'll go into more detail about that as we go. But those are the 2 primary parameters that we'll focus on tonight on the fatty acid testing. So to start out, in terms of milk fat, 98.5%, 99% of the fat is a chemical structure called triglycerides. A triglyceride has a glycerol backbone, and there's 3 fatty acids attached to that structure. That is 98.5% to 99% of the milk fat. The rest of it, there's some cholesterol, there's phospholipids, some of the minor lipids. The 3 fatty acids that are attached to the backbone can vary in chain length, the number of carbons, all the way from 4 carbons up to 18 and longer in terms of number of carbons. These fatty acids then can be different chain lengths on each position, and then they can also vary in the degree of unsaturation or number of double bonds in the chain. Those are variations in the structure of the fat that actually, what we're finding, with the infrared analysis, we can measure those differences in structure, and that's some of what we're going to talk about. So the triglycerides are kind of our main target in understanding their composition and structure as it relates to how the cow is interacting with her feed and how that changes with stage of lactation of the cow from when she's in negative energy balance to positive energy balance. In terms of unsaturation, here, these structures, this would be linoleic, the fatty acid with 2 double bonds. They're cis double bonds at the 9 carbon and 12 carbon. It's 18 carbons, and the colon 2 means that there's 2 double bonds. And let's see if I can get the pointer here, maybe. So here we have a double bond and a double bond. And one of the things you see, these are 3 fatty acids, linoleic acid. This is conjugated linoleic acid. It's got a cis at the 9 carbon and a trans fatty acid or trans double bond at the 11 carbon. This is 1 double bond and a trans. You can see from the pictures here that with a trans fatty acid, the chain is relatively straight. Oh, you have a laser pointer. Okay. So here is at the 9 carbon, a cis double bond puts a bend in the chain. and another one at 12 bends the chain even more, so the structure is not straight. With a single trans double bond, the fatty acid chain is relatively straight, like a stick. If it had no double bonds, it would be totally straight. These change the melting point of the fat and will influence the fluidity, how liquid the fat is, in the mammary tissue itself. So the cow needs to keep the fat liquid, and we'll use these differences in chemical structure of the fat to keep everything working and flowing correctly. So these fatty acids are in the structure of milk fat, and these would be the unsaturated fatty acids. And they're important because in the rumen, the dietary fat, particularly corn silage and a lot of the oilseed type products, have a lot of linoleic fatty acid, the cis-9, cis-12. The rumen is a chamber where biohydrogenation, because it's relatively anaerobic inside the rumen, occurs, and the normal process at normal rumen pH is that this unsaturated fatty acid would be converted to CLA, cis-9 trans 11, and then would be converted one step further towards saturation. So this is what's called biohydrogenation to the 18-1 trans 11, and ultimately, if it goes all the way, it converts it to a saturated 18 carbon fatty acid. This is the normal process of biohydrogenation that's happening in the rumen when everything is working properly and rumen pH is where it should be. And these fatty acids can travel through, if we do this right, these fatty acids, as we go through the biohydrogenation process, then are transported via the blood and ultimately go into the mammary cells. And this is normal. We have trans 11. Some of it can be transformed to CLA, to the cis-9 trans 11. And now if I go and put my food science hat on, when a consumer consumes milk fat with trans 11 C18-1, in their liver, they can actually transform, a human can transform this fatty acid to this one, which is the real desirable one from a nutritional point of view. So all of this is normally happening in terms of the fatty acids in the rumen and hydrogenation. But if this level of the fatty acid load in the rumen becomes too high, and other things start to happen that influence rumen pH, we get into a situation where the biohydrogenation happens differently and has some negative effects in terms of milk fat depression. And that's what we're focused on here in this slide. Here we have the same dietary linoleic acid, and this is the main fatty acid that would be in corn silage. So there's quite, in corn-based dairy diets, there's a lot of this fatty acid there. And when the rumen pH is too low, the microbial population is a little bit different, and this hydrogenation process goes a little bit differently. And what happens is that we get the formation from the same linoleic acid. We get biohydrogenation, but we get a different form of CLA. We get a trans 10 instead of a trans 11, and we could get some stearic acid. But these two are the ones that are the issue. That when those are transported through the blood and get to the mammary cells, that these specific fatty acids inhibit what we call de novo fatty acid synthesis. And I'm going to talk about that and exactly what we mean by de novo fatty acid. So these are inhibitors of the normal process of taking low molecular weight substrates and building milk fat in the mammary tissue, and we're going to focus on that next. So here, to define what we're measuring, so these are the tools that I made for the infrared. The de novo fatty acids, the infrared analyzer gives an answer for de novo fatty acids in grams per 100 grams of milk and can calculate the relative proportion of the total fatty acids that are de novo. So we can express it in two different ways. What's included in the metric that we do is from C4, 4 carbons, butyric acid, all the way through the 14 carbon with 1 double bond. These are measured as a group. This group of fatty acids are made in the mammary cells from low molecular weight substrate, and we're going to talk about that next. The mixed origin includes the palmitic, C16-0 and 16-1. They're about 35 to 40% of the total. The short-chain fatty acids, or the de novo fatty acids, are 18 to 30% of the total. And then what we call preformed, these 18 carbon and longer cannot be made from scratch from individual low molecular weight compounds, cannot be synthesized in the mammary cells. So they come in preformed from the blood. They can either be mobilized from adipose tissue, or they're fatty acids that came in the diet and go through and get processed in the liver and come to the mammary tissue and go into milk. So in general, expectations are going to be 30 to 45% of the total fatty acids in milk fat. So that's the big picture. And instead of focusing on every individual one and trying to figure out this one's going up, that one's going down, these relate to different origins. So this mixed origin, they can either come preformed, already made into the mammary cells through the bloodstream, or we can make them from 2 carbon units that are coming into the mammary cell, and we'll talk more about that, and elongate it 2 carbons at a time to form a 16. So that's why we call it mixed. It could come from either of these sources. So de novo fatty acids, we feed a cow, we get a rumen fermentation, particularly the fermentation and digestion of forage to produce volatile fatty acids, VFAs, and the two that are of real interest for milk fat synthesis are the acetate and butyrate that are produced in the rumen. They are carried through the bloodstream. This is a 2 carbon and 4 carbon unit. They're carried through the bloodstream to the mammary cells, and those substrates are put together in a biochemical process to form fatty acids of the various chain lengths from C4 up through 16 carbon fatty acid and come out in milk fat. So as a group, the de novo fatty acids are of interest to us, but they're not something that are being eaten and in the diet from the cow. They're put together in the mammary gland from the substrates that are from the fermentation. So if the fermentation isn't producing enough acetate and butyrate, de novo synthesis is going to slow down. In terms of the preformed, cow eats feed. There's fatty acids that are longer chain fatty acids in the feed. Those can go through the rumen, go into the lower digestive system. They're absorbed, and they can be transported with lipoprotein carriers to the mammary gland, where they're incorporated directly in the triglyceride structure as preformed fatty acids. In a transition cow, so while the cow is in negative energy balance, the adipose tissue, the fat deposits, are primarily long-chain fatty acids, palmitic, stearic, the 18-0, 16-0, 18-1 to some extent. When the cow is in negative energy balance, she'll mobilize some of that fat. That's carried through the blood and also supports the synthesis of triglycerides in the mammary tissue. So these two categories are the key thing in understanding the balance and how they change in response to days in milk and how the cow is interacting with the feed. So our first objective was to develop some analytical tools. We've done that. The second is to determine how to use those milk fatty acid composition data on bulk tank and individual cow milk samples for feeding and health management in dairy cows. And we're going to start with the bulk tank and talk about that. And I'm going to share with you some data from a large study that we've been doing over the last 4 years with the St. Albans Co-op in St. Albans, Vermont. What is interesting with this technology, they had the machine, the instrument, and they were using it routinely for payment testing to measure fat, protein, and solids. What I did was enable the same machine with no additional work to give more information on every sample. Doesn't take any longer to do the testing, but it gives you more information. And we started producing fatty acid data every time they did a payment test on every farm in the co-op, and started looking at and trying to understand how we could use that information. So here I'm going to share with you some of the data. This is the data from 15 months worth of testing for 430 farms, bulk tank. Every farm is tested a minimum of 3 times a month. Some of the larger farms as many as 20 times a month in terms of bulk tank. This is percent fat on the y-axis, and this is de novo fatty acids in grams per 100 grams of milk. And one of the first things as we started doing this and looking at the data, because no one had data on a large group of farms over a long period of time to understand if there was any relationships between these different fatty acids and bulk tank fat test that's different from one source of fatty acids to another. So one of the first things that we saw very clearly, now the range of fat all the way from below 3 up to nearly 5%, 4.7, 4.8, this reflects in the 430 farms both Holstein herds and Jersey herds. So that's why the fat content goes high when we get into the Jersey herds. The interesting thing is to look, the R squared is about a 0.6 in this case, population data, 15 months. The slope of the regression line about 2.16. And as we move next to look at the Jersey farms, we see the same relationship, about the same slope, maybe a little bit higher, but generally the de novo fatty acids, as they go up in concentration, the fat test gets higher. And you can say, well, if you put more of any kind of fat in milk, it'll get higher. So we need to look at all of them and do they behave the same or differently? So this is the de novo fatty acids. If we look next at the percent fat, actually this seems like we're flipping over to unsaturation, and this is the metric that I said in the beginning that relates to the overall level of unsaturation in the milk fat. This is de novo fatty, or this is double bonds per fatty acid. So if you had population data with a census and you talked about the overall number of children per family, and the average for a country is 1.4 children, and you'd say, what's 0.4 of a child? Well, this double bond metric is kind of the same thing. How do you have a fatty acid with 0.3 double bonds? You don't. We're looking at triglycerides, and if the metric says 0.3, it approximately means that 1 out of the 3 fatty acids on the triglyceride has a double bond. That's what it means in terms of the metric. What we found is that in general, as the double bonds per fatty acid increased, the fat test went down. And we observed that, but we really didn't understand what it was telling us. And as we go along, I'm going to share more detail in terms of what these things mean. This is the same thing. We saw the same relationship in Jerseys, that as the milk fat became more unsaturated, the fat test went down. when we look, this is protein. This was interesting because in the beginning we didn't look at protein, but I had some time and started looking at the data. What we found is the bulk tank protein test increased as the de novo fatty acids increased in the milk. And at first I needed to talk to some of my animal science friends to say, so why is this happening and does it make any sense? So why did the protein increase with increasing de novo fatty acid production in producing the milk fat? As we have more de novo fatty acids produced, it's really telling us that forage digestion is working very well. Our VFAs produce in the rumen, the volatile fatty acids, the acetate and butyrate, is strong. We're producing a lot of it. We're providing a lot of substrate for milk fat synthesis. And the fact that that rumen fermentation is working really well and producing a lot of the volatile fatty acids, it also means that the microbial population in the rumen is very good, that we've got a big biomass, microbial biomass in the rumen, and that biomass represents a supply of essential amino acids. So that we always see this, that there's a correlation that if we do things in the diet to be able to support and promote the production of de novo fatty acids, we also see that it promotes the production of a higher amount of protein in the milk. This is the same thing in Jersey farms. We saw exactly the same trend. And this is interesting in Jersey farms. So these are some Jerseys that weren't doing so well down at this level of de novo fatty acids. And for a Jersey farm, this is really low protein. But you see that ones where everything is working very well and their diet is right, rumen fermentation is right, it's nearly 1% higher in protein test. This is bulk tank. These are whole herds. So this is pretty big differences. And the R squared on this is 0.7. So, and this is population data. This is not a confined research study. This is what the herds are doing. And this is off a lot of tests over a 15-month period. So in terms of the overall from this study, the conclusions from the preliminary work of the 430 farms is that as de novo fatty acid concentration increased in grams per 100 grams of milk in the bulk tank, we had fat and protein concentration increase in the bulk tank. As the double bonds per fatty acid in the milk fat increased, or the level of unsaturation, the fat and protein went down. So we knew this, but we didn't know anything about how much milk was being produced per cow. So when the concentration of fat and protein went higher in the bulk tank, was that just reflecting the fact that the cows were producing less milk per cow and the total grams of protein and fat output for the day really wasn't any different? We didn't know the answer to that question. So we moved forward to do a follow-up study in more detail to understand and try to get an answer to that question. And that's what we're going into next. There were two studies, they're field studies. In each year, in 2014 and 2015, there were a group of 40 farms in each year that we went and visited the farms. We sorted all the data from the 430 farms, and in the first year, 2014, we looked at the Jersey group of herds, the Holstein group of herds, and what we did is we selected 10 Jersey farms that were low in de novo fatty acid production and 10 that were high. So we had 20 Jersey farms, and we did the same thing with Holsteins, where we had 10 Holstein farms of low de novo and high de novo. And what we wanted to do was go to the farms and try to understand what are these farms doing differently that influences their fatty acid composition and influences their bulk tank fat test and protein test. In 2015, we did basically the same thing, except from our learning in the first year, we decided to go in the second year and do all Holstein farms to focus more on Holstein farms and understand the relationships with de novo fatty acids in Holstein farms specifically. So going into the first study in 2014, this is the data for each group of 10 farms. So there were 10 Holstein farms here, low de novo, 10 high, same thing with Jerseys. And this is data for these herds prior to the study to look at the differences. So in the Holsteins, the ones that were higher de novo fatty acids, the fat test was nearly 4% versus the ones that were low at 3.6% fat. The protein was basically 0.15% fat, percent protein higher. This is in the Holsteins. When we look at the Jerseys, similar trend in terms of differences, but the differences are much larger. In the Jersey cows, it's nearly 0.9% difference in fat tests between the low and the high. And I do a lot of research and work with cheese factories. This 0.6% protein is a huge impact on cheese yield. What are these Jersey farms that have this kind of test doing different than the low de novo ones, and what's the production per cow? If we express the results in grams per 100 grams of fatty acids, which is also a useful way of looking at this, that the low de novo Holsteins are about 24% of the total fatty acids as de novo, and when you get all the way to the Jerseys, you can see the progression. You get up to the average for the group of 27. You'll see some farms up in the 28 to 29 range. And on the low end, on the Holsteins, you'll see farms down at 21 or 22 on the low end. And these other values, since they've got to add up to 100%, will also change in harmony with that. So this is going into the study in 2014. We've selected intentionally groups that differ in de novo fatty acids. What I'm going to show you is for the Holstein farms in 2015. The detail here we've got, and seems like maybe we skipped a slide. Yes. So here, this one is percent fat and de novo fatty acids grams per 100 grams of milk. And basically what we're finding is that if you want on a Holstein herd to be a bulk tank fat test of 3.75% or higher, then the measured grams of de novo fatty acids per 100 grams of milk is going to need to be higher than 0.85 grams per 100 grams of milk. This is kind of the cut point that if you don't reach this, you're kind of going to be stuck at a lower fat test. But what's interesting here is this data, these are Holstein herds. Holstein herds with between 4 and 4.2% fat. And we're at 0.9, 0.95, nearly 1 gram per 100 grams of milk on the de novo fatty acids. So this is the range in 40 farms that were in this study, and we selected a group of low de novo and high de novo, and you see the R squared is 0.8. So this is a pretty strong relationship in terms of the relationship of this metric of fatty acid composition to fat test. Here, if we take the de novo and mix, because part of the mix comes from the de novo synthesis, part comes preformed, the R squared, 0.88, and basically the cut point is around 2.25. If you're not there, if you're lower than that, you're going to be lower than 3.75% fat in the bulk tank. And you can see here that as you move this sum of the two up, you're going to go up in fat test. What if we look at the preformed fatty acids? So one of the things is the variation in this across the herds wasn't really as large as the other fatty acids that were present. These do not come from the rumen fermentation. And basically, there wasn't much relationship of the preformed fatty acids, the 18 carbon and longer, with fat test, and the R squared was 0.06. So in terms of trying to achieve higher bulk tank fat test and protein, the de novo fatty acids seem to be a really good metric to understand whether things are working correctly in terms of promoting milk fat synthesis by forage digestion or not working very well. This is double bonds per fatty acid. This is the unsaturation of the milk fat and percent fat. And again, we saw in these 40 farms, basically if you want to be higher than 3.75% fat in the bulk tank with Holstein cows, the double bonds per fatty acid is going to have to be less than 0.31. And when that goes higher, fat test is going to go down. This is in the 40 farms. If we look at this on Holsteins, I've seen Holstein herds up as high as 0.4, and usually their fat test is down around 3 or 2.8. And you'll see some Holstein herds get down as low as 0.25, and they'll be up at 4.2, 4.3. Jersey herds tend to run up in this higher range, and the Jersey herds will get down to 0.21, 0.22, and hardly will ever go above 0.31. So this metric, we're trying to understand, we think it relates to, when this goes up, we think that it's, and you're getting high number of double bonds per fatty acid. This is a R squared of 0.7 in this population, that the roof load is either too high, the rumen unsaturated fatty acid load, or the release of that fat in the rumen is happening too easily and too much fat is being released in the rumen. And we think that this metric is telling us something about that. So in relation to milk fat depression, what's interesting is that the formation of trans fatty acids, that incorrect biohydrogenation, when this value gets high, the double bonds per fatty acid, and we look at the de novo are headed straight down, and the R squared is 0.85. This is in herds. This is not something where we were doing a feeding study. This is in the field in terms of what we see. So we're starting to understand that this metric of the unsaturation is a very interesting number that we can produce off the infrared on bulk tank, and we can do it on individual cow milks too. When we look at protein in this versus the 430 farms, as the de novo goes up, the protein percent goes up, and we think this relates to the rumen fermentation and biomass produced in the rumen. And basically at this point at 0.85, if you're going to hit a 3.1 protein or higher in Holstein herds, you're going to have to be over 0.85 grams per 100 grams of milk in de novo fatty acids to achieve that. But we have some herds, Holstein herds that were in the group, that were up in this range of 0.9 to 1 gram of de novo fatty acids per 100 grams of milk. And these are Holsteins, true protein, not a total nitrogen number, true protein at 3.25 to 3.3. So overall, in the study from year one, we selected the two groups of farms to be low and high in de novo fatty acids, and so they were. We expected that. But what was interesting is this is the first time we had milk weights. The high de novo, so this is the high concentration of fat and protein, actually were producing more milk per cow per day, in addition to having the higher components. So now we're really starting to say this is not just changes in concentration. So in the year one study, this is the difference in fat test average for the groups. This is the difference in true protein. Now remember, these are Jersey herds, half of them, and half Holstein herds. The MUN is about the same, the milk, urea, nitrogen, and it's at a reasonable level. And it looks like we're missing the bottom part in this slide. Next slide. Okay. No. We lost the economics part on the bottom. We'll go to the next one. This is year 2, and it's the same story. And let's see at the bottom in the end. This was similar on the previous slide, but the numbers were different. We took this data and took the differences for fat and protein at the production level. In the second year, with the higher producing Holsteins, there wasn't any difference in milk output, kilograms of milk per day per cow, but still the concentration was higher. So those cows were producing more grams of fat and more grams of protein per day from the feed they were consuming. The MUN was about the same. And then we did a calculation of the economics of that in terms of the income per 100 cows. And in this study, it was approximately $15,000 per 100 cows per year in terms of the difference between the high and low de novo herds. The previous year, the difference was larger because
Dave,
the milk. Yes.
I'm going to get that for you. If you want to talk about this one, I'll get that slide up for you.
Okay. So we did that, but the main message in both years was basically the same, that actually when you've got higher de novo fatty acids, you're producing either the same amount of milk or more milk, but you're producing more fat and more protein output per day. So you get from what you're feeding, you're getting more output per cow. And the numbers, they're positive and they're pretty significant when you think about this. Is not for the whole. If you had 1,000 cows, you multiply these numbers by 10. So this is something that I think needs to be focused on. These, in terms of the management practices, in general, the high de novo herds had lower stocking density. They were 10 times more likely to have 18 inches per cow or more. Stall stocking density was lower, so they were less crowded. There was less crowding at the bunk, which would, when they're overcrowded, which was the low de novo herds, tended to be more overcrowded. You get slug feeding and you get rumen and pH going down. And again, that works against the good fermentation and production of milk fat. So we have several of these metrics as we go through, the feeding frequency, and those types of things are in the slides. But all of these are standards that we've told producers: don't overcrowd cows. The feeding frequency needs to be appropriate. And all of this came out in the field study of what we're observing is that farms that were following the recommendations generally perform better, and in relation to push-up and stuff like that. So also the herds, the high de novo herds, tended to have a higher physically effective fiber, the PENDF. They were 10 times more likely to have a value of 21% or higher. And this is more of Heather's bailiwick, but it relates to the form of the feed. It was interesting, the low de novo herds, the diets contained more dietary fat. It wasn't a big difference, but it showed up both years that there was more fat in the diet in the low de novo herds, but neither of the averages were excessively high. And this is where I'm starting to wonder whether the quality of the fat and the release of the fat was different in the different groups. And in the future, we'll look more at that. Factors related to de novo fatty acid synthesis: less bunk space per cow is related to lower de novo, lower fat, lower protein. Higher stall stocking density, not enough resting space, they're standing too much. lower de novo, lower fat, lower protein, higher average fat, ether extract in the diet, lower de novo, and higher PENDF tended to favor the milk production. This is overall for Holstein herds, and I have to emphasize this for Holstein herds, that if you are less than 0.8 grams per 100 grams of milk of de novo fatty acids, you're pretty much destined to be lower than 3.8% fat. If you're less than 23% of the total fatty acids as de novo, you're going to be down 3.8 and lower. Here's the cut points for mixed origin. And the fatty acid unsaturation, if you're over 0.31, generally you're going to be lower than 3.8. And you could make a similar type of thing for protein. They're correlated with each other. So the main conclusions, the strongest correlation between milk fatty acid composition and concentration of fat and protein was with de novo fatty acid production. When double bonds for fatty acids increased, milk fat and protein decreased, and it seemed to be clearly indicating that there was some level of milk fat depression happening in the herd. The de novo fatty acid level seems to be a barometer of rumen health and proper rumen function. And this is a very interesting metric that we can tell right from the milk and get an overall average for the herd. So feeding and farm management strategies that produce an increase in the synthesis of de novo fatty acids may produce an increase in milk fat and protein percentage and really possibly more production per cow per day in terms of those high value components. So even more information is gained when we start looking at this on individual cows, and I'm going to give you a little insight of what we're doing there. One of the things that we've done in addition to fatty acids on individual cows is typically in transition cows, when they're mobilizing too much body fat and we're worried about ketosis and displaced abomasums and other metabolic diseases, we take a blood sample and look at the non-esterified fatty acids, the NEFA. So one of the questions I had is, could I test milk and give you an estimate of what the blood NEFA test would say without ever taking a blood sample? So this is something drilling deeper on individual cow milk samples into what do we see and what can we predict. So our objective was to develop and validate a Fourier transform, that's the infrared, the FT, mid-infrared based milk analysis system, to measure NEFA concentrations or estimate NEFA concentrations of the blood by testing milk from lactating dairy cows. So in the transition cow, we've got the calving going from negative energy balance to positive energy balance, this high risk period during the first as long as 10 weeks, and there's all kinds of things that we can measure, but generally if we want to get a fat mobilization measure, we take a blood sample and do NEFA. And the question is, can we do that from milk, and can we get as many of these metrics as possible from milk and integrate them into deciding what the risk is and what to do in terms of minimizing ketosis or displaced abomasums particularly. So the NEFA concentration measured in the blood represents a concentration at an instant in time when you take the blood sample. The levels when you measure blood NEFA on blood samples will vary with the time when you took the sample in relation to feeding, varies with level of stress of the individual cow at the time of blood sampling. And if you're repeatedly sampling blood from cows, they tend to get more nervous when they see the person coming with the needles. So one of the things that's interesting, it's hypothesized that the blood NEFA concentration estimated from the milk represents a time average status of the blood NEFA for the full period of time between milking and no stress. So it's a different number. You wouldn't necessarily expect it to agree exactly with a one point in time blood test, but it might actually give you an average of what's happening in that cow. So we think, And we're starting to see that these numbers from milk are much more stable, and we're watching that across time in individual cows and trying to get more of a sense of how to use it. This is an example. So what I want to do is explain a little bit about this graph. So on the y-axis over here on the right side, this is de novo fatty acids, and the scale goes from 10 to 26. The x-axis is days in milk, so calving over here at 0, and now we're in the high-risk transition period. And on the left side, this y-axis is the blood NEFA. And generally, we want the blood NEFA less than 700-800. When it's up in 12 and 1400 territory, the cow's mobilizing too much fat too fast. So this is the blood NEFA estimated from the milk, and these are the de novo fatty acids. So the blue line is the de novo fatty acids. This is the estimated blood NEFA, and this is an example of a cow with high body condition. She's got quite a bit of body fat at calving, but she seems to have good liver function, and her blood NEFA starts out higher than we'd like to see it, but it comes down pretty rapidly in the first few days, and she's coming down nicely. And this is what is new when we have this de novo fatty acid measure, that what we're seeing is a cow at 4 to 7 days usually starts out with about 20% of the fatty acids as de novo. And if everything's working right, that climbs and should get up to 26, 27, maybe even 28% of the total fatty acids as the cow approaches positive energy balance. So she's making more de novo fatty acids. Her preformed are decreasing. So this is a cow where everything's working right. The next slide. This is a cow that actually had a displaced abomasum. So her blood NEFA started out high. She was coming down, but what's very different about this cow in measuring the fatty acids at day 4, day 7, day 10, instead of her de novo fatty acids being about 20 and going up, they're going in the other direction. It's going down. She's not synthesizing the shorter chain fatty acids, and basically this cow had a displaced abomasum. At this point, which was about 16 to 17 days, she had surgery for the DA, and after the surgery, what was interesting is very rapidly the de novo fatty acid synthesis recovered, and she started coming up as you'd normally expect. So in this cow, the surgery worked. This was a left DA. This next one, this is the same sort of graph, and this is a cow where the de novo was at 20 at day 4, came up a little bit, and then it just kind of didn't change. And what was interesting is her estimated blood NEFA wasn't coming down like it should. It actually went up a little bit, and here glycol treatment was started. She was headed towards ketosis and actually was diagnosed with clinical ketosis. Once the glycol started, her blood NEFA started to come down, and the de novo fatty acids came up. So you can see in the milk that it's working. So these are new metrics from milk that we think will be useful in individual cow health management. So the milk estimated blood NEFA and fatty acid data correlated well with documented ketosis and displaced abomasum, but we've got a lot more work to do on that to really develop a strong base so that we understand where the cut points are to be able to test the milk and decide what action to take on that individual cow and when to take it. Future directions, we're really going to focus more on the blood chemistry measures. We're working on the blood BHB. Everybody has the MUN, but can we do stress and inflammation compounds, things related to reproduction? They'll be useful in understanding these. And one of the questions we've got and we're working on is, from the milk, could I be able to test the milk and tell you something about what's happening to rumen pH? So those are things that are in process now that we're working on. I don't know whether we can do it, but we'll try, and we'll see what we can achieve. Took a lot of people to do all this. The folks at St. Albans Co-op, Minor Institute, all are part of the team. Delta Instruments, the equipment company, giving us technical support. The United States Department of Agriculture Federal Milk Market Labs working with us and helping fund the methodology improvement. And that is what we've done. And Fourier Transform Infrared rules. It's the technology, the testing technology. And I'd be happy to answer any questions.
Okay, thanks, Dave, and nice car. I'm going to take over control for a few minutes and do some of our closing details, and then we'll open up the floor for questions. So before I open up the floor, I want to invite our listeners to attend our final webinar of the year to be held on the 9th of November at 5 p.m. Eastern Standard Time. We will be hosting Dr. Nina von Keyserlingk. Dr. Marina or Nina von Keyserlingk is professor of Natural Sciences and Engineering for the Research Council Industrial Research Chairholder in Animal Welfare at the University of British Columbia, and is recognized internationally for her research on care and housing for cattle. Because of this work as an educator on farm animal care, the impact of her research can now be seen on farms around the world. Save the date and time on Wednesday, November 9th at 5 p.m. Eastern Standard Time. She will be talking about rethinking methods for the feeding and housing of dairy calves. We want to thank the people who make this possible. They are Tom Taluki and AMTS USA and Global, Marcos Neves Piera, University of Lavras, Marcelo Hens Ramos at 3R Lab in Brazil, Paula Turillo in Argentina, as well as our translators in each location. Our generous sponsors make it possible for us to get great speakers and manage the program. We thank our gold sponsor, Ajinomoto Heartland, superior nutrition through amino acids. Our silver sponsors are Arm & Hammer Animal Nutrition, Virtus, makers of Strata with EPA, DHA omega-3s, and Prequel with omega-6s. Bronze sponsors are Jeffo, Life Made Easier, Dairy One Forage Laboratory, and Dairyland Laboratories, and Quality Liquid Feeds. I will now open the floor up for questions. Please type your question in the question and answer tab, and I will get to them as I can. I try to take turns. And also, we're going to welcome Heather Dann to assist in any nutrition-related questions you might have. So, Heather, hello and welcome. How's your mic?
Hi, good afternoon, good evening.
My first question that I'm going to ask is from Steve Halleroy. First of all, he said thank you for a good seminar. And he had a question on the units for fatty acids you used. Grams per 100 grams of milk, not percent total fat. Is there any specific reason for that?
The reason is that the instrument itself is actually measuring and calibrated to grams per 100 grams of milk, and that's what the model is predicting. We also predict the total grams of fatty acids, and we can express the grams of de novo fatty acids as a percentage of the total fatty acids. Those would be the numbers like 23%, 25% of the total. But we're starting to understand that for taking action in management, I think the grams per 100 grams of milk is going to be more useful. And that's been our experience in the field, and we're starting to focus more on that. And Heather may want to make some comments on that also.
I think Dave explained it very well, so I don't have anything to add at this time.
Okay. Steve also had a follow-up. Do you think calibrations for individual fatty acids would be possible?
I think that they are possible, and we've done that. We have calibrations for some individual fatty acids, but I find that, one goes up, one goes down, and looking at it from the perspective of a group of fatty acids that relates to the biosynthesis pathways of how things are happening tends to give me more useful insight into what's happening in the biology of the cow and how she's interacting with her feed or how she's changing that ratio of fatty acids in production in relation to days in milk, her energy balance status.
Okay, thank you. I'm now going to have a question from Paula.
Okay. Hello, Dave. Thank you very much. I have a
You're
question
welcome.
from Eduardo from Uruguay. Do you think differences in milk fat concentration between AM and PM milking are due to differences in de novo fatty acids?
We have not looked at that, although we have AM/PM milking data at the Minor Institute, and Heather may be able to focus on that. But in general, when we see differences from AM to PM, the question of the length of the time in the milking interval, if it's twice a day milking and it's not 12 and 12 hours, then I'd expect some difference in the balance of how much is produced in each milking. And also the behavior of the cows, that different individuals are different when you look at individual cow milk. And some cows tend not to eat much during the night. It just depends on differences. And the sort of temporal pattern of how they consume feed on an individual cow basis can influence that fat concentration, actually the amount of de novo synthesis. The resting time, rumination time is also important. And that's kind of where I'm at. Heather may have some insight into the AM/PM because they're looking more closely at individual milkings from the same cow across time.
Yeah, so that's a great question. When we look at our bulk tank data, we will definitely see differences in the time of day of milking, in the amount of fat that we have in that bulk tank, and then the makeup of that fat is different as well. And so we will see a difference or a higher amount of de novo fatty acids in, for example, our midday milking. We milk 3 times a day, along with higher total fat percentage in that milk compared to when we look at those same samples first thing in the morning or later in the evening. And so we're trying to understand better why we see those big differences, whether it's total fat percent of the milk or the individual components. And I think a lot of it goes back to probably our feeding and management behavior of the animals. So when they choose to potentially eat, especially under heat stress conditions here at our farm, we saw some changes this summer. We'll continue to see some changes. Maybe under certain stress situations, they're going to change, and that'll be exacerbated. And as mentioned before, milking interval may have a role as well.
Okay, thank you, Dave and Heather, both. I'm going to have Paula ask another question, and then we'll go to Marcos.
Okay, thank you, Heather. This is a question from Elvio. You said the level of double bonds are related to corn. Could you tell us a maximum recommended level of corn feeds in our diets?
That would probably be a better question for Heather.
Okay.
Yeah, so I don't have an actual guideline for corn. What I'm more focused on is the fermentable carbohydrate load in those diets and how that's going to impact rumen function and therefore the biohydrogenation intermediates and fatty acid flow that will eventually end up in the mammary gland and can influence milk fat. So in general, in high-producing herd diets, we're going to be less than probably 30% starch. NDF maybe around 28 to 32, depending on the quality and digestibility of that fiber. And so we can achieve those levels with a number of different sources of carbohydrates. So I don't focus as much on just the absolute pounds or kilograms of corn that's needed. The other part of that story is looking at fermentable carbohydrates, but also paying attention to the amount of rumen unsaturated fatty acid load, or RUFLs, that may be provided in the diet. And RUFL is really going to be the sum of the primary unsaturated fatty acids. So we would look at C18.1, C18.2, and C18.3. And typically recommendations from Dr. Jenkins at Clemson would be that on a diet dry matter basis, we'd want to be potentially less than 3.5%, or at least use that as a cut point to determine if we have a higher or lower risk of having some milk fat depression. For many herds, that might equate to about 600 to 700 grams of ruminant unsaturated fatty acids.
Okay, thanks, Heather. I think if people want to refer back to Dr. Jenkins' presentation that he gave in May, he may have covered that. I'm going to see if Marcos has any questions.
Yeah, we do.
Okay, great.
I have another question for Heather. She said that the fatty acid profile is higher in the afternoon, the midday milking. But when you talk about fatty acids as a percentage of the total, does it change a lot? I understood that you said about the fatty acid as a proportion of the milk. But does it as a proportion of the fat? Do you have experience with that?
Yeah, so what we'll actually see is, we've looked at it both ways, grams per 100 grams of milk and also grams per 100 grams of fatty acid. And we will also see a little changes between or among those different milkings, but when we graph it out visually, we see less of a difference when we express it as relative percent or grams per 100 grams fatty acid. So that's one reason Dr. Barbano and us were encouraging people to start to look at this information on grams per 100 grams of milk. We think that's going to relate more positively to changes in milk fat and milk protein and be able to interpret that in a more meaningful way. So what's essentially happening, they're probably changing maybe their feeding behavior due to maybe some management or environmental constraints or issues that then probably have an impact on the total amount of, for example, de novo fatty acids being produced. And the ratio when they're being produced is staying relatively similar. We do see a little variation, but that's smaller than the absolute amount.
I could give an example. If you take a cow and intentionally restrict its feed intake and watch what would happen to fatty acid composition, the thing that you'd see, so in terms of percent fat, grams of fat per 100 grams of milk, that would get down, but you'd see that the de novo and the mixed would probably go down more. And if we were thinking about this as relative, the relative percentage of the preformed would go up, but it's not that they're producing more preformed fatty acids, they're producing less of the de novo and the mix that's coming from the de novo synthesis. So when you start thinking about it of how the two would react, you can get a higher preform, not by them actually making or putting more preform in the milk. If the de novo and part of the mix goes down, the relative proportion of the preform goes up. So that's why sometimes it's a little hard to interpret what's happening when you're looking at the relative percentage of the total fatty acids, because it's always got to equal 100%. And when you look at the grams per 100 grams of milk, if the de novo is going down and the preformed is staying the same, you kind of can better understand something's influencing the de novo synthesis. So that's an example where if I withhold feed, but now if I'm feeding cows and something has changed in the ration, so the energy density of the ration isn't as high as it should be, it probably will produce an effect like restricting feed intake, and you're going to see the de novo go down, and the preform is going to stay the same in grams per 100 grams of milk, but at a relative percentage, it's going to appear to increase.
This question is from Alejandro Urbina from Costa Rica. He said, you mentioned limit on appropriate ether extract content of the diet. Does the source of the ether extract make a difference?
Yeah, we can tag team it.
Yep.
So we've looked in the survey work we've done on 2014 and 2015 on farms with the St. Albans Co-op, that we see that herds that have higher de novo fatty acids as well as total fat percent tend to be farms that feed a lower fat or ether extract. In those herds, we saw a difference around 3.5 to 4% fat in the diet. That's kind of low compared to what I think sometimes we think about feeding to our cows, where we might be closer to 5% and hopefully typically not exceeding 6% of the diet dry matter. And then along with just a total amount of ether extract or fat in the diet, we need to be aware of the makeup of that fat and the unsaturated fatty acid load in the form, and looking at the sources of those fatty acids. So are they coming from our forages and our cereal grains and byproducts, or are we incorporating some rumen inert or bypass fats into the diet as well to minimize the negative impact of any unsaturated fatty acids on the rumen environment and the microbes?
Okay.
The only comment I'd add to that is one of the things that doesn't seem to be captured in any of the feed testing is two feeds with the same amount of the unsaturated fatty acids. And in total, if you analyze them, they're the same. But if they release that fat differently in the rumen, it could have a different effect in terms of milk fat production. And from a feed and forage analysis point of view, we don't have many tools that really tell us anything about that. And it could have an effect in terms of the rumen fermentation.
Okay, thank you. I have a question. This is from Alberto Navas Camacho, and this is about grazing system. His question is, under grazing systems, we supplement cows twice a day with 3 to 4 kilograms per cow each milking time. Concentrates may have 5 to 7% fat. Do you think this fat load in such a short period of time, for example 3 minutes, could be more deleterious?
Yeah, so I guess I would think first of what that form of the fat is and how available or not it's going to be in the rumen, as well as the other components of that diet. So I'm assuming, given that makeup of 3 to 4 kilos of that containing, or that grain, a fair bit of starch and potentially some sugar. And so I think you need to look at the profile. As far as the feeding behavior and the impact of that, I can't answer that directly of if that's going to have a bigger negative impact on the fatty acid components. But to me, I think of it more along the lines of if we were in a confined system or out on pasture, as you described, that it's more of a slug feeding approach, or you're going to have a larger fermentable carbohydrate load at one time, and potentially that could have some impact on the rumen and therefore have some interactions with the fat sources depending on what they are.
Yeah, I would tend to agree that I think that the question of how that's going to influence the rumen pH, and that may have as much of an effect in terms of how the fat gets handled as the level of fat itself.
Yeah, so I think the point about low rumen pH is key, because we know in situations where we have lower rumen pH and a large supply of unsaturated fatty acids, that's going to increase our risk for milk fat depression.
Okay, thank you. Go ahead, Paula.
I have a question from Elvio. What do you think, I think this is for Heather, what do you think is more important regarding milk fat depression, total fatty acids or low rumen pH or acidosis?
Yeah, so it's a great question. And I think one of the challenges that we face with milk fat depression is that it's not just one factor that's going to have an impact often. It's not going to be the interaction. And in severe cases, we may see one or more of those factors being obvious cause of milk fat depression. But really the combination of lower rumen pH and total fatty acid load that's even, maybe close to what we might normally feed or on the upper end we're going to see some negative impacts. So I think we have to evaluate both of those as well as some other factors on farms that are going to influence milk fat depression, and some other things that I think about when I'm looking at diets or thinking about the feeding environment is, how rapidly available is that starch and fiber coming back to the point of acidosis, but also then potentially the use of ionophores and what role they may have, as well as what role yeast and mold may play in some of the ensiled forages or grains that may be feeding, and the negative impacts that they can have on the rumen environment.
Okay, thank you, Heather. The last question from Danielle. Would you make some comments about seasonal impact on milk fat depression?
I'm going to answer this one. I know you've looked at a lot of this over time.
Yeah, we see seasonal variation in terms of fatty acid composition and milk fat depression in some of the herds. When we have those 430 herds, you'll see some herds cycle like that seasonally and others don't. And we want to try to understand that. One of the things that in some of the herds that cycle, the question of the changes that are occurring in the silage, particularly corn silage, as the silage ages across the season, and particularly in all the regions that I work with, that typically we see in the spring, before there's any pasture, that the composition of the milk starts to change, fat and protein start to go down. But one of the things that we've been watching now is that when that's happening, the temperature outside, the environmental temperature, is increasing. And we're beginning to wonder whether that sort of triggers and promotes more things happening in terms of the silage as it warms up in the bunker storage, and it changes the availability of starch, which I think we know, and Heather knows a lot about that, but also the question of whether there's some changes in the structure of the fiber-fat interaction in the silage and changes in the release of fat from the structure in the rumen when the cow consumes it. And to me, if something changes the way the fat comes out of the forage in the rumen and increases that, the fat load and the fatty acid composition is the same, but if you just pour in more pure fat free, negative things are going to happen. And that's what we're starting to watch, and we're starting to think about where they have their silage and what the environment is around that and the temperature it's exposed to, if it's in the shade versus it's in a location where it gets a lot of sun and heats up quite a bit, what impact that has on the release of fat. So we're suspicious that some of the seasonality relates to changes that are happening in the silage type of feed as a result of the environment that it's in and its condition. And Heather may want to comment on that too.
Yes, I think, Dave, you made some good points there, and it's definitely an area that we need to follow up more, and it seems to be one that may be a culprit on some of our farms where there's not obvious causes of the lower milk fat or milk protein, and one that we definitely need to look at further.
Okay, thanks. I have another question from Alessandro Urbina. If you need to feed bypass fat, what would be the ideal fatty acid profile for that supplement?
Yeah, so I think the specific fat, there's lots of different rumen inert fats available on the market, and I think we want to use one that's a good quality product that is going to deliver those fatty acids post-ruminally, and we need to look at the digestibility. And so quite often those fatty acid sources are going to be higher in palmitic acid, as well as some of our essential fatty acids, so the C18 fatty acids.
I want to Dr. Barbano. Dave, thanks for the presentation. It's about the technique. Someone is asking how the curves will be available, if a commercial lab can have it, or if you have any prediction on that. And also, I think you can answer at the same time about the precision of the equipment. For example, we're talking about changing the de novo proportion from 24 to 26%, and seems to be a really minor thing, and how the machine is sensitive to this little change.
Okay. The first part of the question, the models that we've developed in terms of these statistical models actually have been done in collaboration with Delta Instruments. So the models that we've developed and we're using in the field, actually the same model runs on their machines. And this year, they're introducing in their software the de novo, the mixed, and the preformed will be available to customers. And probably in the next group of things that are released, they're going to, I think, release the blood NEFA and this double bond per fatty acid. So those are going to be commercially available on the Delta Instrument equipment. We're publishing papers on that. There's, it takes a lot of hard work to develop the models, but there's no reason why other brands of equipment can't develop similar things. It's just that it takes some work and understanding. So those are available and they're being released on some of the equipment. The second part of the question in terms of the performance of the models, it's a very good question. In terms of from an analytical metric point of view, often the precision of a model is expressed as coefficient of variation. The de novo fatty acid measurement analytically of the fatty acid measurements is the best. And that's fortunate because that's a real important one. The coefficient of variation in a external validation study that we did is around 2%. And to put that in perspective, the fat testing method we use for payment is about between 0.5 and 1% coefficient of variation. So it's not as good as payment testing, but it's a relatively good method. And methods that have a coefficient of variation between 5 and 10 are considered qualitative decision-making methods, and above 10% coefficient of variation, those are fairly weak models. So in general, and this is being published in a paper that should come out in Journal of Dairy Science, that these metrics are in that publication. And all three of these, the de novo is about 2% coefficient of variation, and the other two are up around 4 to 5% coefficient of variation. Hopefully that answers your question.
Oh, yeah. Thank you. Yeah, it's on the same topic. You made a decision to use groups of fatty acids. I think someone has asked something about analyzing individual fatty acids. Have you tried to work with ratios? For example, I saw the correlation when you add the mixed fatty acids to de novo, you increase the correlation a little bit from 0.8 to 0.88, I think. For example, could you use the ratio of the mixed with the preformed, or even measuring single fatty acids like the trans 11 and trans 10 and using a ratio of those, things like that, other metrics. Probably you tried.
We have been trying that, and in general, when you look at these individual fatty acids, some of them, the metrics in terms of analytical performance is much better than the others. And to give you an example, I do have a model for the C18-1 trans 10 and the C18-1 trans 11. That's a very difficult distinction to make analytically with an infrared analyzer for two reasons. One, the structures are almost identical, and two, the concentration of those things you're trying to measure is really low. So you've got two things working against you. If we look at, so now those metrics that I just spoke to you about in coefficient of variation, so if you ask me what's the coefficient of variation on the trans 10 and trans 11, between 20 and 25%. I wouldn't bet the farm on that one. The analytical performance of those methods is pretty weak, but in general, it gives me some hope that we might be able to produce better methods on that, but I don't think they're really at the analytical quality at this point that we'd want to be making decisions on them.
Okay, thank you.
Okay, and going back to bypass fats, would you make a recommendation on the percentage or grams of added bypass fat?
Yeah, so typically when I'm thinking about fat for a high-producing lactating cow, I'm going to total fat be maybe 5 to 6% max on a diet dry matter basis. Typically, we're going to have close to 3% of that fat coming from just the basal ingredients, so the forages and grains and that. So depending on the fat source, 1 to 3%. And so each of the specific enterprise products have their feeding recommendations at different times. So, typically, quite often, a quarter to maybe a pound of those products. But it'll depend on, the type of cereals, for example, or byproducts that you may be using, and the profile that those have on how much additional fat you would want to be adding to the diet.
Okay, thanks, Heather. And Eduardo, he's asking, when you have low milk fat, where does that energy the cow is saving go?
Quite often we'll see that fat is going to be diverted to body condition in those animals, especially some of our later lactation cows.
Yeah, I guess the other thing I'd look at is that if the de novo fatty acid synthesis is lower because we're not getting a good rumen fermentation and not producing the load of VFA and the biomass, that we're actually just not digesting as much of the feed components and absorbing nutrients from that, so we've actually got a lower efficiency of digestion and conversion. Maybe it's just coming in one end and too much of it's going out the other.
Yeah, that's a very good point, Dave.
Okay, you can see we have milk fat depression problems here in Argentina. The last question from Pedro. Do you have any information relating milk fat depression and low reproductive performance, specifically pregnant rate?
I don't have anything specific on that.
Yeah, I don't either. It's an interesting question as far as milk fat depression and pregnancy rate. Quite often, at farms that may be having some management issues, it wouldn't be unlikely that maybe the reproductive program could have some challenges as well. But there's been some recent Journal of Dairy Science articles that have tried to look at some specific fatty acids in milk and using those as a index of whether cows will eventually go on it to become pregnant. And there's mixed information, and so it doesn't seem to be, I think it's, an area we need to do further work in, one that we're exploring now, the role of how we can use this milk information to look at, for example, when a cow is either going to come into heat or potentially what is the likelihood that that cow is going to become pregnant once a farmer chooses to breed her. And so indirectly we can use the milk to get some assessment of energetics of that animal that may then be tied into the success of that breeding and milk composition. But as far as low milk fat, I don't have a good answer for you.
Yeah, I think on the breeding and reproduction, to me, if we can understand more about the energy balance of the cow, that if coming into the first cycle the cow is still too far into negative energy balance, I think the probabilities of success in breeding are much lower. And what I'd wonder from the milk is whether we can actually identify a cow in terms of making a decision that based on data and experience of pregnancy rates at first insemination, that a cow that has this characteristic profile that relates to energy balance, it has a very low probability of getting pregnant. That may be something that would be doable in terms of interpretation of the infrared results on the milk.
All right. Thank you for those questions, Paula. Did you have another one?
No, I'm done.
All right. I'm going to
Thank you very much.
Thanks. I'm going to go to Marcos. Marcos, did you have more questions?
Yes, I have a couple here.
Very good.
It's about feed management. What feed management practice would be more correlated with the de novo concentration of fatty acids in milk? And at the same time, you showed a negative relationship between pushing feed and the fat acid, the fat test, and then the de novo concentration. Could you develop a little bit on that? You think the guys that are pushing feed, they're feeding grain or hot diet or something?
I guess my first reaction is that whether it's managing the way feed is presented to the cows and in relation to the crowding the cows, that anything that produces a lower average rumen pH or more cycling of rumen pH going low is going to have a negative impact on de novo fatty acid synthesis and milk fat production, milk fat test. So that would be the first thing I'd be thinking about. And that's where it's interesting to me on milk analysis. If I could get a metric that told me something about how the rumen pH was changing on average for the herd from a bulk tank test, and if I've changed something in feeding for the whole herd in terms of a source of silage or something like that, and I see the whole herd move down in terms of rumen pH, I would be concerned about that. And that's where trying to understand how to get metrics from the milk that might give us those insights into how the cows are interacting with their feed.
Okay, thank you.
Along those lines, I think also in the survey work that we've done, we've seen that herds that have higher de novo feed more frequently, and that is an important piece. And you asked the question, I don't think we went over the slide, but for feed. But we saw in our work that herds that had lower de novo actually pushed up feed more frequently. And at first look, it seemed like that wasn't what we would have expected. And what we think may be occurring there is that those farms with lower de novo fatty acids, they're probably, and we saw that they feed less frequently, and because of that, there's probably a need to push that feed that they do deliver once a day or so up more frequently to allow those cows to have access to that.
Oh, that's interesting. Okay. I have another one here, is about the relationship when you look at the data from Jerseys and from Holsteins. Jerseys seem to, like the fat test and the protein test, seems to correlate much more with the de novo than with Holsteins. Do you think we should be using Jerseys more on research to study milk solids? Like we always use Holsteins because farmers use a lot of Holsteins, but Jersey seems to be better models. Can you comment on that?
I'm looking for eligible Jerseys. I've found a university herd, not at Cornell, but another one, where I'm trying to get some things started to actually get more individual cow data on Jersey cows. I think it's an interesting question, but we don't have much data on that.
Okay. Another question. You have any experience with using this data for transition cows only post-calving? Like, for example, you see a lot of potential for these metrics to replace like the milk to fat to protein ratio, or things like that. I think it may be, seems that data on the sick cows and the fresh cows seems to point in that direction. What do you think?
Well, I think what we've seen that really makes a difference. Now, you can look at that fat-protein ratio, and it does tell you some things. But what we're finding is the metric that's the blood NEFA metric, estimated from the milk, I think is much better than looking at fat-protein ratio. It's really starting to tell you more about mobilization. The next test that we're working on is to actually predict the blood BHB, but predict it from the milk. And I think once we get that, what will be interesting is to look at how the two of those move together may tell us or give us some insight into liver function and how that individual cow is handling the mobilization of the fat, and whether the liver is working very well or whether the liver is not able to keep up. So I think some of those things will be very useful and maybe give us more understanding of metabolically what's happening than just looking at fat to protein ratio.
I have the last one here. There's a comment that the solid composition of the forms you worked with was really high for our standards, at least for confined cows. And do you have a lot of data on low fat and really low fat and protein herds, and the behavior would be similar if you have it?
In what we've seen, usually the lowest fat herds are just a little below 3% on Holstein. On Jerseys, we get down to 3.5, 3.4 on fat. And in general, what we see when we look at the metrics is they all fall on the same regression line, that it seems to be the same thing happening that's influencing the lower fat level. It's just that it's more extreme in terms of what's happening, whether it be rumen pH or biohydrogenation, particularly when we get to those very low levels of fat and protein. Usually the double bonds per fatty acid, you start to see that number at 0.35, and I've seen some herds in the 0.4 and higher. Those herds are way down. They're usually at 2.75 to 2.8 on true protein, and the fat test is 2.8, 2.75. So they all seem to follow the same rule. It just seems to be the severity of what's happening.
Okay, I think I'm done. And thanks, Dr. Barbano. Congratulations for the work, and you and Heather. I think it's very important, and you'll get there. Thank you.
Yes. Thank you, Marcos, for your great questions. I don't have any more questions on my end, and Paula has indicated she doesn't, and I think that we can all agree that we were really grateful to have Heather Dann join us, and Dr. Barbano gave such a great presentation. Thank you, Dr. Barbano. Thank you, Heather Dann, and Paula and Marcelo and Marcos for all the help on this webinar. We will see everybody next month.